Upwards, rising from the ashes like the phoenix once again to celebrate science and nature on Fridays. So for those of you who are not familiar, there are two premier journals in the science world, sort of like the Super Bowl of science, and they are called Science and Nature. And if you're wondering what the difference is between the two of them, well, it's the Atlantic Ocean. Nature comes out of the UK, and Science comes out of the US, specifically out of New York City. And for the last 19 years, I've dedicated about the first 15 to 20 minutes of every one of my classes, regardless of the class, to covering the highlights of what was published in these two journals, because anyone taking a science class should have the opportunity to learn about all the cool stuff that was published, well, this week. All right, so without further ado, let's jump into science. I'm gonna pick science first because there's some cool stuff in here on COVID-19 that is worth sharing, but we'll get to that in just a second. So what do we got first on that? Ooh, here we go. Oh yeah. Have you ever wondered, does all that screening at the airport really matter? What does the data show? It shows that it makes us feel better about ourselves, but it doesn't actually catch very many people. What's the actual number? About 1 out of 46,000 travelers that were tested, only one was actually found to actually have COVID-19. Although I guess there is something to be said for making us all feel better. That is part of public health management for sure. Okay, ooh, this is really interesting. So interesting. There's a nice review article in here on sickle cell disease, which is caused by a single point mutation in the beta globin gene. And so should represent an easy target to fix. And so that is moving forward. So there is, in the EU, they are now approving for beta thalassemia, so similar disease, taking stem cells out of patients, fixing the point mutation in those individuals, and then putting the stem cells back into them and reversing the disease ideally. So that's a good thing. All right, here we go. If you're in the third quarter like me, living in your 50s, hopefully you haven't experienced this, but if you've had a herniated disc, you may be aware that if you have a discectomy, which I did not have, but if you have a discectomy, the probability of having a reherniation or disc degeneracy is not improved by the surgery. And why is that the case? Or can you improve it? Turns out somebody did something very simple. They injected hyaluronic acid as well as cross-linked collagen into the disc that was being repaired, and at least in sheep, in the 6 sheep that they tested, so not done in humans yet, it seemed to matter. Now, sheep don't live as long as we do, nor do they, well, I don't know, who knows, maybe they do tax their spine as much as we do. I do not know, because I'm not a sheep. All right, what else have we got going on in here? Oh, let's just cut to the good stuff. All right. So here's what's known. So actually for all viruses, they have actually I don't know if it's all viruses, but certainly for COVID-19, they have what's called a spike protein that binds to at least 2 receptors in human cells, one of which is called ACE2. And the structure, this is science at its best, the structure of the COVID-19 spike protein has been solved, and that is what it looks like. So for those of you who are not used to looking at these things, the little candy cane things, those are called alpha helices. There's some beta sheets in there, which are like little folded sheets, and then some amorphous stuff. Of course, that doesn't really mean much to anything but a protein chemist. So let's look like what it might look like in a more informed way. So this is actually what the spike protein looks like. There's it right there. Where's my finger? Right there. And that is the target inside of human cells. And if you look, the pieces match. So that is just beautiful structure function analysis right there. Of course, why should we care? The real reason we should care is because if you know how the pieces fit together, there's the potential for disrupting how they come together. All right, let's move on to nature this week. I'm going to come back to the cover here in just a second. Actually, why don't we start with the cover? The cover of Nature this week is a piece of amber. It's actually not that large. I think it's only 31 millimeters across, but it has a 99 million year old bird skull inside of it. It's like a miniature predatory hummingbird-sized bird, and it's now the oldest known fossil from the Mesozoic period. That's cool. You don't see that every day. All right. You know, if it's too good to be true, it probably is. Nothing's for free in life, right? So as we're all aware, some very clever chemists / biochemists came up with artificial sweeteners. They taste sweet and they don't have calories because we can't digest them. And for a long time, it's been thought, or actually now it's been known for a little while, that perhaps they're not good for us because nothing is for free. So someone did something very simple experiment. They just took people and they gave them sucralose, which is an artificial sweetener, and then they gave them sucralose with a carbohydrate, or sucralose alone, sucralose with a carbohydrate, or just the carbohydrate alone. And then they measured insulin resistance and changes in response to insulin. And guess what? If you're taking an artificial sweetener and eating carbs, which you're probably doing, your body is more likely to develop insulin resistance than if you just ate carbs straight up, or if you went to an all artificial carb diet, which I don't think you can actually do the latter. Anyway, just one more example of how you really can't cheat. Oh, okay. This may be the most important paper of the day, actually. So this is looking at susceptibility of children to coronavirus. And the question is, are children as susceptible to coronavirus as adults? Dramatic pause. The answer, yes. Doesn't mean that they get as sick as adults do. The mortality is very clear. It's very, very low in children, but they get infected at the same rate as adults. And that is a huge paper, as you can probably imagine, because that very much affects public health and the question of, should we try to prevent it from getting in children? Because can they or can they not get it? Yes, they can get it. Will they get sick? Odds are not. Odds are in their favor. Can they spread it? Oh, yeah. Okay, there we go. That's a big paper. That gets you into the Super Bowl of science. That's why you're in Nature. Okay, another paper in here looking at, and the question is, why does the coronavirus spread so easily? This is really interesting. So the that spike protein that I showed you that was published in Science seems to bind to the ACE receptor about, what did they say? About 100 times more efficiently than the SARS spike protein binds to its receptor. Okay, so it binds with higher affinity. Higher affinity means it's going to be able to stick longer and infect cells at a higher frequency. Interesting enough, they did find an enzyme that is required by the host. So we actually have to provide an enzyme to cleave that spike protein for the virus to invade our cells. That enzyme is called furin. And are you ready for this? Furin is found in, wait for it, lungs, liver, and small intestine. And guess where the coronavirus can infect? The lungs, the liver, and the small intestine. Structure function followed by cause and effect. Ooh, here we go. Don't you love it when science fiction actually becomes reality? So CRISPR-Cas9, the gene editing tools that we've all heard so much about over the last couple of years, they've been used successfully by taking cells out of people, engineering the cells, and then putting them back into people. What hasn't been done up until this week, or actually they're just reporting on it this week, is actually taking CRISPR-Cas9 and putting it into a human and having it fix the genetic lesion in that individual. So this is a type of blindness that is called LCA10. It's a rare form of blindness, actually, caused by a single gene. And what they're doing with this CRISPR-Cas9 cassette is they're putting it into people's eyes, literally through eye drops. That's why you can do this. That CRISPR-Cas9 delivers this payload, and it goes in and actually deletes the mutant form of the gene, and then ideally it's going to restore vision. We'll see. They just started editing in clinical trials now. Keep your fingers crossed. All right, if you're like me and you want to read the latest, greatest published papers, peer-reviewed on COVID-19, there is now an open source hub called Lit COVID, for literature COVID. You go to Lit COVID, they have all the papers from all over the planet curated in one place. One-stop shopping, get all your primary lit. Here we go. Here we go. God, I love biology. I really do. I really, really do. All right, pathogens. Pathogen shows up. In this case, I'm going to talk about bacterial pathogens. They're very often there to feed. In fact, that's what they're there to do. So you look at MRSA, methicillin-resistant Staphylococcus aureus, or just Staphylococcus aureus. When you get a staph infection, one of the things the bacteria does is it produces a toxin, secretes this toxin. This toxin gets into the membranes of your cells and it makes a pore. And then all the good stuff, like the juice out of your cells, comes running out and the bacteria feeds on it. So it turns out that our immune system, our cells, when they're under attack by a bacteria like Staphylococcus aureus, produces these vesicles called exosomes, which then they secrete out into the environment around them. So as the bacteria is trying to insert these pores, they actually, at some frequency, will insert pores into these fake cells. They're like little sponges or targets or decoys to try to fool the bacteria. And clearly at some frequency they work because we recover from infections. And sometimes they get overrun, which is when we have to go to the doctor and get some antibiotics. All right, here we go. Another paper on COVID-19. I'll just let you read the title there. A pneumonia outbreak associated with a new coronavirus, probable bat origin. There we go. More data to suggest that COVID-19 transferred from a bat into humans. All right, that's all I got for you this week. I look forward to seeing you next week and maybe in between on some more COVID papers if I find some that are worth sharing. In the meantime, remember, let the crisis bring out the best in you and rise like the phoenix from the ashes.